we're going to go over the experimental genetics today, the approaches that we use to study development. Really, the whole purpose of these is to answer three big questions. So let's start with experimental embryology. There is no genetic manipulation in experimental embryology. All it is you are doing things to the cells themselves. You're either killing the cells or moving the cells around to find out how development works. And there are a number of things that can be answered with experimental embryology. And I'll give you several examples for each one of these experiments on how it applies. So let's look at the first one, defect experiment. This is one where you destroy cells, and then you see what happens. When you destroy these cells or a portion of cells or whatnot, then you want to see, well, what effect does that have on development overall? So here's a prime example of that. This is a frog embryo, Xenopus, as we call it. And in its first stage of cleavage, where it's dividing into two cells, what they'll do is they'll kill one of the cells, but not remove it, because that does actually change what happens afterwards. So they'll kill this cell and just leave it there. What they find is that if development continues, only half of the embryo will develop. And this cell, or this portion, this huge portion, will pretty much remain as it is. So what question does that answer? Well, when we talk about development, a lot of times we're going to talk about how, yes, there are factors in the cell that tell the cell what to become, but it also matters its environment. So what the defect experiment tells us is, what does the cell-to-cell communication have to do with development? And as you can see here, it has everything to do with development. In order for this cell, it just doesn't say, oh, that one's dead, so I'm going to become the entire embryo. No, it knows that one's dead. It's communicating via various proteins and signals that knows that that half is dead, and it only makes half of the embryo. So this is a conditional state in which it depends upon what the cells are interacting with in their environment. And that's one of the things we learned from the defect experiment, is that cells, their interaction with one another during development matters. It's not just about the genetics. Okay. Second one is called the isolation experiment. This experiment asks and answers the question whether or not a cell has the potential to become every possible cell in an organism. One of the prime examples of this is the sea urchin. What's fascinating about sea urchins is in the first stages of development, if you separate these 4 eggs, they all have, or these 4 cells, they all have the potential to become an entire sea urchin. They all have the potential to form into that larvae and then metamorphosis into the sea urchin. So the isolation experiments basically, and you do this periodically, you can do it at different stages of development, but it tells you whether or not those cells have the potential to become any cell in the organism. Later on, as you get more and more of these cells, you start removing these cells, sometimes they're like, oh, well, that only becomes muscle now, or that can't become the whole organism itself. So that's what we learned with the isolation experiment, is at what stage of development do these cells stop having the potential to become every cell in an organism? Here, as in the earliest stages, that retains that potential. In fact, in human embryos, we know that in the situation where you have lots of cells and forming that inner cell mass, if that inner cell mass splits, you get identical twins. You get monozygotic twins, which are genetically identical to one another. So the potential in those initial cells is there to become every cell in the body, even in human embryos. Okay? We don't perform these isolation experiments, but that is an example of a naturally occurring process where cells get isolated from one another and can become an entire embryo itself. All right. Recombination experiment. This is also one where we ask ourselves a similar question to the isolation experiment. At what stage do the cells lose their potential to take on any fate, any cell type? So in a recombination experiment, especially with the frog, the Xenopus, What they do is they'll take cells from one portion of the embryo, they'll move it to another portion of the embryo, and then they'll see what happens. Well, if you take it early enough, you can see that, again, we do this through some type of either fate mapping, where we can watch the cells and what they become. And that's what we learned about in lecture 1. So we know we can follow these cells and know exactly. We kind of pull them out, we put some dye in them, we put them back in, and then we see where they end up. So what this is showing is that at this stage of development, all of these cells up here, when they're put into this environment, they take on that new state, meaning they have the potential to become cells of the belly rather than the back. So they're still in what we call an undifferentiated state, where they have the ability. Now, we'll learn later on that after a certain point, when you take cells and you move them over, that's not always the case. In fact, I'm going to jump ahead real quick to lecture 6 just to show you an example of that. Here's the same thing, where we're taking cells, recombining cells from one part of the frog embryo and putting it into another, but this is much later in the game, so to speak. And guess what happens? When you take these and put it into another part, it doesn't just take on that fate, it actually has predetermined to become what we call an organizer, or an area that can organize the embryo. And guess what the result is? Twins, essentially. Two embryos that will develop as a result of just moving one part of the tissue to another part, and you end up getting, conjoined embryos, so to speak. So these are the questions we ask and answer with recombination, is at what point do the cells stop having the potential to become any cell in an organism? And that tells us that up to that point, that certain factors are kind of, very pliable. And when we start learning how that works, we can learn how to manipulate stem cells so that maybe we can reverse it. And that's really where a lot of stem cell research comes into play, is understanding how they got to that point so we can reverse it and make it so that they have the potential to become any cell in the body. That's the research aspect of this. Okay, and then the last one is the transplantation experiment, where you usually transplant it from one organism to another of a different organism. Recombination typically is within the same organism, but a transplantation, if you think of trans being different, transplantation experiment is one where you take it from one organism and to another. Now, it could be the same species, but typically they are separate species when you do these types of experiments. And I've given you the example before of this. Remember, when we do a fate map and we want to do a permanent marking of the cells to follow them, such as the quail and the chicken embryos, they're so similar that the cells behave. The quail cells behave just like chicken cells for a long time, and then we can use quail genetic markers to find where those cells end up. So transplantation experiments like this typically are used in fate mapping. Again, fate mapping is used to determine where these cells end up. And by knowing where these cells end up, we can start looking at some of the factors that predetermine how those cells are going to become part of the neural tube and part of the brain and spinal column and things of that sort. So those are the 4 types of experimental embryology: defect, isolation, recombination, transplantation. We use this today, not as often, though, as we use genetics, though. Genetic embryology is kind of where it's at. There are about 4 different groups of techniques that we use to study the genetics. And again, when we say genetics, we mean everything that's involved in the central dogma of cell biology: DNA to RNA to protein. It's a universal process in every living organism and in all cells of DNA to RNA to protein. That's what we call the central dogma of cell biology. Well, genetic embryology is basically manipulation at any of those levels. Manipulation of the DNA, manipulation of the RNA, manipulation of the protein. Sometimes the manipulation of it is to add things to the embryo. Sometimes it's to take things away. Sometimes it's just to look at things. You don't necessarily want to mess things up. You just want to look at them. Again, proteins, DNA, and RNA are so small that we typically have to have tags, some type of marker, dye, a fluorescence, or something to allow us to see it and visualize it and localize it, so to speak. So that's really kind of the main things that we do with genetic embryology. We either take genetics out, we put things in, or we just look at things. Okay, so these are all of the various techniques. We look at where the RNA is found. We look at where the proteins are found. These are kind of one and the same because some techniques are designed to look at the RNA, and as a result, we're assuming, it's a big assumption, and sometimes you have to verify that, but we're assuming that if the RNA is made, so is the protein. We talked about last time, though, that's not always the case. You may have RNA without actual protein translation occurring at that particular time. So typically, RNA is, they're done in conjunction, but there are pros and cons to each one that we're going to go over today. Now, if we want to permanently modify a cell, then we can insert DNA into that. This is when we can add genetics. Now, again, when adding genetics, it doesn't necessarily mean that we're going to alter anything. In fact, sometimes the most common DNA that we insert is just GFP. We just insert some type of protein that fluoresces, that is a transgene from another organism, a jellyfish or whatnot. And there's all sorts of different types of. There's yellow fluorescent protein, green fluorescent protein, and so on and so forth. Blue. But sometimes we do insert a foreign gene into it. Or we take a gene that is not active in one cell, and we make it active in another cell. So there's a number of ways in which DNA insertion can be applied. Now, in combination with that, that's where transgenics and knockout mice come into play. Transgenics typically means inserting a gene into a cell. Knockout typically means silencing that gene in those particular cells. So on the one hand, the transgenics, now that's not always the case. Sometimes we have to do transgenics to knock out the genes. Okay, so it can kind of be both. Transgenics applies to both DNA insertion as well as to knockout. And I'll talk about some of the differences between those two. Because sometimes we can actually put in a system that functions, and then we treat the animal, like a mouse, with a particular substance, and when it gets into the cells, it'll actually cause recombination of the DNA and excise or remove that gene so that it's gone. And the reason why we have to do that is because if you knock out the gene too early, the mouse won't get past a certain developmental stage. So people have created some ingenious genetics that you can put into the organism. It'll function normally. You feed the mouse something that gets into its cells, and then it recombines and removes the DNA at that stage of development, and then you can see what happens. It's just amazing. So transgenics, knockout. We either insert DNA to see what it's doing because it's normally not expressed in that cell, or we remove it, and we want to see what happens when we remove that gene and it's no longer functioning in that cell. And then this last one is similar to the knockout, but it's just another technique where we can actually inhibit the transcription or translation of DNA and RNA. We can selectively inhibit a very particular gene or its RNA to prevent the protein from being made, because that's really where it's at. When we're silencing genes, we're really just removing the protein. When we're adding genes, we want to add the protein. You should know, again, that it's all about the proteins. It's all about the product that gets made and then what effect that's going to have later on to the cell. These are the three universal questions we ask in all of developmental biology. No matter what you are studying, these are the three questions you always ask. Okay? Doesn't matter whether you're studying limb development, whether you're studying muscles, neurons, any stage of development, These are the three questions. So what are they? Well, the first one, when we study the genetics, because we're looking at the mechanics of how the cells are becoming skin and neurons and tissues and whatnot, we first have to ask a localization question. Is the gene expressed at a particular time and place? We want to know where and when that gene is being expressed. What is the goal of this? It's basically just to look at the natural process of development. We're not manipulating anything at this point. Again, this is the detection question. This is answered by RNA and protein localization, typically. These are the questions that, or this is the question that you use RNA and protein localization techniques to answer. Time and place. Where is the gene expressed? And how long, and so on and so forth. Those are all the things we want to know, because the more we know about when and where it's turned on and turned off, because that's what happens. Sometimes genes aren't always on. They get turned on and off at different stages. That's the first question we have to ask. And then we ask the next two questions. Once we know where and when these genes are being expressed, then we ask the next two questions, which is that gene necessary for the development of those cells and tissues? Meaning, if you remove that gene, will the cells still do what they're supposed to do? Will they still become neurons? Will they still become muscle? Will they still become, bone, whatever the case may be? So that's the necessity question. Now, how do we do that question? We typically remove the gene. We get rid of it. We knock it out. We prevent it from being transcribed or translated. So that's where DNA inhibition and RNA inhibition come into play. Or the knockout mice. Or, this is the typical, we call them knockout mice. You can knock it out in any organism, but knockout mice are fairly common in research because, especially in adult mice research where we're looking at the effect that drugs have when they have a particular genetic mutation in the adult. I worked with them all the time when I was at Scripps. And so that's why we just say knockout mice. But in reality, it's just making sure that the gene is not expressed and then seeing what happens. That determines necessity. So getting rid of its expression will answer the question, is that gene necessary for those cells to become that particular cell type? And then the other question we then ask is sufficiency. Let's say it is necessary. Then we want to know if that gene alone is responsible for the development of those tissues, or are there multiple genes involved in that process? So sufficiency is usually done by misexpression. And when I say misexpression, I'm talking about DNA insertion. We add DNA to cells that normally do not express that gene, and we make it so that that gene is active in those cells so that they're expressing the protein, and then we see what happens. So that's the sufficiency. We typically take a gene and activate it in another cell type. We usually do this through a plasmid construct that has a universal promoter that is in every cell, so that there's no repression, there's no selection, it's just on. We just make that protein in that cell type. So sufficiency, is the gene by itself sufficient enough to promote that differentiation, to tell the cell to become neurons? Okay? And that's done by adding it in and making sure that it's active in those cells that it's normally not active in. And there's a number of ways of going about doing this. Again, if you think about, let's say you didn't know anything about a car, and you wanted to know something about the radiator. You knew about the radiator, but you didn't know what its function was. So guess what? You pull it out, and you realize it's necessary for cooling the car down and all these other things. You remove it, and you see all these problems that occur about it. And then you ask the other question. It's a little bit harder with the car example to look at sufficiency, because you'd have to, do a number of other things. But that's really where a lot of teratology comes into play as well, where we see natural mutations, and then we see something happening. We're like, oh, that gene was mutated. It's necessary for this. All of a sudden, we realize that necessity. So the question's already answered for us because nature mutates the gene, makes it unusable, and then we see some result, some teratology, some birth defect that comes about because of that gene mutation. It's a little harder to do sufficiency naturally, so to speak. But okay, so northern blotting. It's just a way of determining where RNA is. And that's typically done through gel electrophoresis and a membrane, and there's a number of ways in which you can detect it. In some cases, the way in which this is done outside of the cell. So you actually take a group of cells that you're wondering if the RNA is actually made in those cells, you grind them up, you run them on a gel, you put them on a nitrocellulose membrane, and then you do some detection methods to see if they're actually there. So that's a northern blot. For looking on whether or not an RNA is there or it's not there. It's a localization. But it doesn't tell you specifically the exact location of the RNA in the cells. We can actually use other techniques to do that same thing. Reverse transcriptase PCR, this is one of those things that is the reverse of PCR, where we're actually making probes. That's just a process to be able to make probes that are used in northern blotting and in some of these other techniques. For example, if I want to make a probe for a particular gene, extract the RNA, and then I'll do reverse transcriptase PCR, which is just taking the RNA and making a cloned DNA copy of it. Microarray. So an RNA profile shows you, it's a hybridization process where you actually have known RNA strands that hybridize to other RNA that you're looking at. The great thing about microarrays is it gives you hundreds of RNAs at a time. It's not like a northern blot where you maybe can only detect one type of RNA at a time. A microarray can actually tell you whether certain RNAs hundreds of RNAs are found in a particular group of cells. So it's a little more expensive. They are getting cheaper to do. When they were first invented, you had these little chips, these biochips that you use for them, and they cost $40,000 each. They're less expensive now, and they're used more in research than anything. But that's a microarray. It's a similar process to a northern blot, except it can profile hundreds of RNAs being expressed in a particular cell at a time. Because sometimes you need to know that profile to know how the proteins will interact with one another. It's just very tedious to look at one RNA at a time. So that's a microarray. I won't get into the details of all these little dots and what they mean and everything, but ultimately it's showing you whether or not the RNA is there or it's not, and intensity levels and things of that sort. Okay, another way which we can look at where the RNA is found, another localization of RNA, is called an in situ hybridization. Now, there are huge advantages to in situ hybridization over northern blotting over a microarray, because in situ means that we can actually look at where the RNA is expressed in the whole embryo. We do not have to remove the cells to be able to see where that RNA is at. That's a huge advantage in development to discuss not only if the RNA is being expressed, such as the time frame, but the place in which it is found, which we discuss is a huge question that we have to ask ourselves. So how is it done? Similar to a northern blot, you have probes that are designed to attach to the messenger RNA. So the same thing is true in a northern blot or a microarray. There are typically RNA probes that hybridize, which just means complementary base pairs, to the RNA in question, the one that you're interested in finding. Well, in situ, this actually does this hybridization in the cells. So you have to treat the embryo with a number of chemicals to make them porous and allow for the RNA to come in. This process takes 3 freaking days. May not seem a long time, but it is, especially when you mess up somewhere along the middle and then you don't realize till a couple days later. So I've torn my hair out doing quite a number of these. Sometimes they can look nice and clean, like this one right here. Other times, this one's okay. It's actually pretty good compared to other ones that I've done. But I didn't do this one right here. This one's immaculately clean in terms of that. But what an in situ does is it hybridizes the RNA to where it's found in the cells, and only the cells that RNA is expressed. Then you go through a process where you have some antibodies that recognize a chemical that you've added to your probe. And those antibodies attach only to the RNA, because you wash this and you get rid of all of the RNA that's not bound to any of the RNA in the cells, so that only this complex will form in the cells that have that RNA. That antibody typically has an enzyme, an alkaline phosphatase, attached to it. And when you add substrates such as chloronaphthol and whatnot, it will cause the clear solution to precipitate out and form a purple substrate that will not leave the cell. As such, you get this beautiful staining where you can see exactly which cells are expressing that RNA. You can see these are actually what we call neural crest cells. You can actually see the cells migrating from the neural tube down into these regions. It's just amazing. This is one of the cleanest in situs I've ever seen. But and a lot of it depends upon how good your probe is and everything. You're assuming, which is a big assumption, that proteins are being made where that RNA. And that's usually a good assumption. It typically has been verified by other experiments that, yes, the protein is also made. But if no one's ever done these experiments before, you have to typically verify it yourself to make sure that there is that correlation. Otherwise, it's a big assumption being made, because sometimes the RNA is there, but not the protein, and that makes a difference. Okay, here's another example of immunohistochemistry, or of how you can use kind of the same technique as you would do for a Western blot to see something, but this is actually using fluorescent antibodies. This is a very good technique. Here you're seeing the cytoskeleton, you're seeing the filaments as well as the microtubule, and then you have the nucleus. I believe these are pseudocolored. The nucleus is typically done with like a DAPI stain, which is blue, but it's been pseudocolored red as a contrast between some of the others. You can get anywhere between 3 to 4 different fields of fluorescence to be able to overlay on a single cell or a single grouping of cells. All immunohistochemistry does is a process of typically either labeling the RNA or labeling the proteins themselves. If you have the antibodies synthesized in a lab, and they're very expensive, which is why not everyone does this, the in situ, that's the poor man's detection, really, or the poor lab's detection. So if you don't have a lot of money in a lab, typically you do in situs. If you have a lot of money, you can have somebody create antibodies that are specific for the protein that you want to find, and then you can use those to detect and actually look at the proteins and where they're found in the cells themselves. Okay, so those are the techniques for localization, knowing where the RNA is found or where the protein is found. Most of these is RNA, northern blotting, microarrays, in situ hybridization. Most of those are just looking at where the RNA is found. Again, northern blotting and microarrays, the advantages is they're very easy to do. The disadvantage is you don't get to see where in the embryo they're actually being expressed. Yeah, if you extract some tissue from the brain, you know it's in the brain. But it's so much better to be able to just see it in its native state in the embryo. And that's where in situ hybridizations have an advantage in our knowledge and understanding of this. And as far as immunohistochemistry goes, that's mostly used for protein detection, mostly used for knowing where the proteins are actually found. And you can do the same thing. You can do what's called a FISH, or fluorescent in situ hybridization, which is similar to in situ, but it uses fluorescence instead. And you can use that for protein staining in the embryo itself. It's as labor-intensive as the in situ hybridization is as well. So all of those techniques essentially answer the time and place. Is the RNA located or expressed at this time? And where? What cells are actually expressing that RNA or protein? Now, DNA insertion. There's several different ways in which we can insert DNA. Some are natural processes, others are mechanical processes, so to speak. And each have their pros and cons. Now, again, for DNA insertion, it can be for two purposes. It can be for adding DNA so that you can answer the question of sufficiency. Remember, when you add a gene and have it expressed in cells that normally wouldn't express that. I mean, the gene's typically there, but we add the gene on this plasmid because the plasmid has a promoter, and the cell will synthesize the RNA and the protein off of that plasmid. So that's the advantage of adding DNA in that way, is we can, what we do, misexpress a protein in that tissue and determine whether or not it causes the cells to become, you know, whatever they're going to do. But there's also, when we insert DNA, we could do it for the purpose of knocking the genetics out. Okay, so we could insert a gene and have it permanently be part of the genome and actually replace the native gene that's in there for various purposes. So DNA insertion is just the process of inserting the DNA, but it can be used for either one, either for knocking things out or for adding things in. More often than not, it's used for adding things in. It's used for the sufficiency question. Okay, so here are a couple of ways in which we can insert DNA. First one is just direct physical injection, a microinjection into the nucleus, where we take a very thin glass pipette and we can actually inject and insert DNA into the nucleus of a cell. It's a little difficult to do, but it's more of just a physical brute force, you know, injected into the cell or directly into the nucleus of the cell. That's typically what a microinjection is. It'll puncture the outer membrane and then go right into the nucleus, and then you'll just inject it into there and then pull it out and hope the cell survives. An easier method, but a little more expensive, is lipid transfection, like lipofectamine. It's a substance that can actually form these kind of lipid micelles, not missiles, micelles, M-I-C-E-L-L-E, micelle. They form these lipid micelles around the DNA, and the reason why that's advantageous is because it can just go right through the phospholipid bilayer. It just gets pulled right into the cell. So the DNA enters almost like a virus. It gets encapsulated, and it comes right into through the membrane and can come right into the cytoplasm of the cell. Another way to introduce it is, and this is what I use for my PhD, is electroporation. You literally pass voltage through the cells, and it does two things. The voltage will cause the cells to temporarily become porous, thus allowing things that are on the outside of the cell to be drawn into the cell through, you know, the water coming in into the cytoplasm and whatnot. You can't fry them. I've fried them before. You got to have, I mean, you're talking about millivolts, just really small, you know, electrical pulses. Now, the advantage too, the second thing is you have the positive end on the opposite end of the cells, and since the DNA is negatively charged, it moves towards that end and is forced into the cell, essentially. Very, very easy way of introducing DNA into the embryo. The only problem is you've got to take quite a bit to set it up so that the embryo is at the right angle. You're setting your electrodes on the opposite ends, and then you've got to use this tiny little glass pico spritzer that just spits a tiny little bit of your plasmid out onto the surface of those cells, and then you hit the voltage and it goes right into those cells. All right, so those are kind of the three main mechanisms of inserting DNA. You can either do it through brute force, you can do it through lipids, where it will come into it, or you can do it through electroporation. When you replace or insert DNA into the nucleus of an embryonic stem cell that will eventually become a mouse, what they do is they'll take this embryonic stem cell, they'll replace the genetic material with some other genetic material that they've already manipulated. Maybe they've knocked out the gene, or they've created a construct so that they can knock it out at a certain point. Well, what they then do is they'll take the embryo, they'll implant it into a pseudo-pregnant female, one that thinks it's pregnant but is not. They'll put it in there, and then it will form the various mice. Well, during the development process, it's kind of a hit and miss in the first generation. Some of the mice all the mice are chimeric. meaning they'll have a combination of different cells. Some of the mice will have in their germline, which is the sperm and the egg and whatnot, will have incorporated into their germline cells the transgenics. So then you breed these mice together, and the next generation you can get mice that have in their entire genome that gene knocked out or that gene inserted into it. So it usually takes 2 generations to make these knockout mice. So here's just showing how they'll inject some of these modified embryonic stem cells into a blastocyst, which is the developing embryo for the mouse. Then they'll inject it into the uterus. Then you'll get these chimeric mice. Now, it doesn't have little lines on them. These little lines just indicate that the mouse is a combination of normal cells and genetically modified cells. There's actually 2 different types of cells that form their makeup. And then you just breed these mice together. Some have in their germline the modified cells, others don't. But what will happen is when you breed them together, eventually you'll get these mice that have both, since we have 2 sets of genes on our chromosomes, have both of the genes knocked out. Typically that comes about as once you can quantify through real-time PCR or something else that the mice are heterozygous, because sometimes they need to be heterozygous because if you knock it out completely, it might kill the mouse in its overall development. So heterozygous mice are used for breeding purposes because they survive, essentially, the development. And then when you mate them together, you can get normal wild-type mice, you can get more heterozygous mice, and the key is the knockout mice, and that's what we use to study what effect it has. Typically, when you knock out that gene, they may have brain defects, they might have limb defects, and that tells us quite a bit about what the purpose of that gene is in development. Another way of knocking out a gene is not actually removing the gene, but creating a mutated version of that gene. And when you create a mutated version of that gene that supersedes the other normal gene, we call that a dominant negative construct. So a dominant negative construct is where you cause a gene to be expressed that's mutated more often than the normal gene, and due to that mutation, it competes for the normal gene's functionality, and it can knock out the expression of that gene just the same. So I'm not going to go through the details of how that's done, but a dominant negative construct is one where you mutate the gene, you put it into the cell, and due to the fact that you make it so that that version of your protein is expressed more often than the native version, then it essentially knocks out the expression of that gene. And then the last concept, in our last few minutes here, is, and this is a fascinating one as well. There's actually a 4th way of regulating RNA naturally that I didn't talk about yesterday. Scientists are finding that within the cells themselves, they actually will create microRNAs. And these microRNAs can be antisense to the RNA that's synthesized. And again, these antisense RNAs sit on the RNA, preventing translation, thereby preventing the protein from being made. So instead of actually targeting the gene itself, we're just targeting the RNA of that gene. It's just another way of getting rid of the protein. What question does that answer? Necessity. Knocking out the expression of that gene answers, is that gene necessary for the development of that stem cell line or that tissue line? So we can, this is a natural process of regulating RNA expression. Now, we can use that to our advantage because we can easily synthesize antisense RNA. So if we synthesize these antisense RNA and put it into the cell, it's the same thing. We're essentially getting rid of the protein, making it so that translation can't occur, and thereby seeing what the downstream effect is of that. So there's actually multiple ways of doing this. I'm not going to go into the details of all of these. The one that I particularly used and liked is the Morpholino oligomers. And let me explain the difference. Antisense RNA has a limited time frame in how often it can be used. When you put it into a cell, what happens is the antisense RNA will typically bind to the RNA, and then you get these proteins that will come along and essentially chew up the RNA. You get double-stranded RNA, and then you get the splicing and cleavage of these, and that essentially removes the RNA from being able to be transcribed. Well, one thing that I like about the Morpholino is it will block translation, but it stays there permanently. So it essentially, every time RNA is made, this doesn't get degraded. It just binds to the RNA, and it can't be broken up by these enzymes that normally cut up double-stranded RNA. So it's resistant to these nucleases, and it's good for a lot longer period of time.